ATP from glucose, you have to memorize the step of glycolysis and Kreb cycle. You have to, guys. I don't know how I can help with that, you know, but there are major enzymes. I'll identify them. At least it will decrease your work by 50%. But nonetheless, the one that I mentioned, there's no way around it. You have to memorize. And guess what? When you guys memorize, you'll be so proud of yourself. And when you see its functionality and its activity in terms of delivering ATP and so forth, you'll be so happy, like you accomplished something. Biochemistry, people dread it. But it's so important. It's such a big deal, because if you can understand biochemistry, everything else will become so easy. I suggest you challenge yourself, like, you know, how dare people think it's difficult? I'm serious. You have to take this challenge. Okay, you have to take this challenge, and you have exam in a week. So for that challenge, remember that there will be about 25 questions, 8 questions from carbs, 8 from lipids, and 8 from amino acid, and then that's 24. One more from one of them, that's 25. 5 questions will be bonus. So even though it will be 30 questions, I'll only count 5. So whatever you get out of 30, it's out of 25. Okay, it is out of 25, but you 30 questions. So if you get extra bonus, try to get 110%. You know, let's see. You gonna get it? Good. Okay. Well, me and him, we made a personal covenant. He came to me. He's like, I'm gonna try to ace all your exams. I'm like, oh, that's beautiful. So that's why I remember I talked to him. But all of you guys should do the same as him. So now, and you succeeded almost, right? Almost. Muhammad Zaghair succeeded too. Good job. Yeah. Nina succeeded too. So that's good job, guys. But try to do it. I'm telling you, it is not difficult to do well. I think it's very difficult to do bad, because it's difficult. It will give you stress, and you'll be like, oh my god, how did I do that? It's difficult, so don't do that. Do the easy part, and that is study and get good grades. All right, let's go with this. So we want to make ATP. Now to make ATP, we have to have, let's say this half is mitochondria, let me see if I can change colors with that. Uh-oh, no good. No good. So I'm gonna make one half mitochondria, the other half cytoplasm. Okay, I just don't have and mitochondria should have 2 membranes, correct? Mitochondria should have 2 membranes. Okay. There you go. Now I can go back. What did I do wrong? I have no idea. Good. Well, hopefully this will work, but let's see. Now, so we're going to start with this. We want to take glucose, break it down so we can take its hydrogens to make ATP. Now remember, I said there's 3 reactions that are irreversible. Glucose being glucose, 6-phosphate, and then this being converted to fructose, 1,6-bisphosphate, and then this one being converted to pyruvate. Okay. If there is oxygen available, and you know this will tell you why oxygen is so important for living. If there's oxygen available, pyruvate can be converted to acetyl-CoA. Okay. And the enzyme for that is pyruvate dehydrogenase. So we need to know hexokinase enzyme. We need to know PFK One, we need to know pyruvate kinase enzyme, and we need to pyruvate dehydrogenase. This is to make ATP in the presence of oxygen when you're breathing. Okay. Although you could, I'll do this on the other side, you could convert pyruvate into lactic acid. LA is okay? Would you remember it's lactic acid? No? You want lactic acid. Lactic acid. And the enzyme for that is known as lactic dehydrogenase. Pyruvate dehydrogenase requires oxygen. I'll put O. This one don't require oxygen. In the presence of oxygen, it won't work. So it actually requires lack of oxygen. So I'll put O with a cross. Lactic dehydrogenase requires lack of oxygen. Okay. Alright. Now, when you're breathing, you're going to take glucose. Convert eventually to what? Pyruvate. And pyruvate is converted to acetyl-CoA. The enzyme pyruvate dehydrogenase is located on the membrane of mitochondria. It's still mitochondrial enzyme on the membrane of mitochondria. Okay. Now, do you remember that fructose 1,6-bisphosphate was converted to glycerol aldehyde 3-phosphate? In that process, we produce, I'll use green for that, we produce NADH. Since there's two of them, so we'll produce 2 NADH from here. 2 NADH. 2 NADH. Okay. Pyruvate dehydrogenase. Anytime you hear the enzyme dehydrogenase, what does it mean? It produces NADH or NAD plus, one of them. They usually can do reverse. So pyruvate dehydrogenase will also produce 2 NADH. Okay. But since the enzyme is there, it can actually do it inside. So we'll just do it this way. It will produce 2 NADH. 2 NADH. Are we good? Why do we care for NADH? Because it's the source of hydrogen. I'll highlight it blue. That's what we care for. Okay. Now, then we have something called the Kreb cycle. Kreb cycle. I'll just draw a circle. Okay. Acetyl-CoA joins right here. Okay. And then many products will go through. So let me just mention the most important products of the cycle. Oxaloacetate, citrate, and then malate, alpha ketoglutarate. That's more than enough. I'll mention only 4 of them, but you guys can memorize all 9 of those reactions. I think there's 9 reactions, right? But I'll mention only a few of them because they're very important. Okay, so oxaloacetate, OAA. Well, I'm running out of room. So oxaloacetate will go here. Here will be citrate, and here will be alpha ketoglutarate and malate. Okay. So oxaloacetate, when it combines with acetyl-CoA, it becomes citrate. Okay. You have this cycle in your book. And citrate is then converted to isocitrate, okay, and then to alpha ketoglutarate. Alpha ketoglutarate. And then alpha ketoglutarate can be converted to many cycle with malate. Okay. Although you have succinyl-CoA, you have fumarate, you have many other steps, I don't care for them. It's a good idea to know, but these ones you should know. You must know. Okay. I should. That's okay. Okay. You know what? I should have included here isocitrate. Sorry, isocitrate. Just include here isocitrate because I think that'll cause us a problem. If I don't put it, let's just put here isocitrate. Isocitrate. So here was alpha. Now this enzyme one here is citrate synthase number 1. An enzyme that combines oxaloacetate and acetyl-CoA is citrate synthase. Citrate synthase. Okay. Citrate is converted to isocitrate, and then from isocitrate you convert to alpha ketoglutarate. The enzyme here is known as isocitrate dehydrogenase number 2. Isocitrate dehydrogenase. Isocitrate dehydrogenase. If it's dehydrogenase, produces 2-NADH from a single molecule of glucose. And then you get alpha ketoglutarate. Alpha ketoglutarate is then converted to malate through many steps. Actually, it's converted to succinyl-CoA, but it's okay. And then to succinate and so forth. But I'll just skip a lot of steps. And in the process, you will have 2-NADH. Malate is then converted to oxaloacetate. Again, 2-NADH are produced. Okay, and our focus is what? On the hydrogens. So this is a hydrogen carrier. This is a hydrogen carrier. This is a hydrogen carrier. Okay. From succinyl-CoA to succinate, you produce 2 GTPs, which is a different thing. I'll talk about it. Okay. Now we have about 2 NADHs produced by glycolysis, and about 8 NADHs produces 6 from the Kreb cycle, 2 from pyruvate dehydrogenase. So we have a lot of NADH. They're nothing more than carriers of hydrogen. That's all. The pump that we talked about earlier, or the channel, the ATP synthase, is located on the inner mitochondrial membrane. On the inner mitochondrial membrane. So look, this blue thing, this blue membrane is the inner mitochondrial membrane. And then there should be an outer mitochondrial membrane. I don't have it, but let me use one more just to show you. So there should be another membrane like this. Mitochondria has 2 membranes. We talked about this day 1. And hydrogen should be accumulating in the intermembrane space to drive this ATP synthase. So once we acquire all of these NADHs, on the inner mitochondrial membrane there is a group of enzymes, and they transport electron from 1 to 2 to 3 to 4 to 5. So they're all in a chain format. Let's do that. Enzyme number 1. Here you go. This is 1. I don't know which page is it on your book. You can 3, 4, and 2. I will make it dark. There you go. And then 5. So these are all enzymes located on the inner mitochondrial membrane. They are known as electron transport chain. Electron transport chain. Electron transport chain. Electron transport chain. Okay? The first one is NADH dehydrogenase. The first one is known as NADH dehydrogenase. If you look in your book, I think the blue box that I see, the numbers should correspond to each complex. Or if there's another box, I don't know. One of the boxes next to the picture. Is it the blue one? Okay. So now what we're going to do is all these NADH, I'm going to take these NADH just to show you. Oh, NADH. So this NADH will come over here. The next NADH will come here. I can keep bringing them, but you get the idea. So what are the total NADHs? 6 from Kreb cycle, 2 from pyruvate dehydrogenase, and 2 from where? Glycolysis. And this one should be shuttled somehow. It should be shuttled. So total of how many? 10. Right? So that's 1, 2, 3, 4, 5, 10. So there's 10 NADHs that will be brought into where? So this NADH will be taken like this. It will be converted to NAD, and the hydrogen ion will be stripped from it and it will be pumped outside. Okay? NADH dehydrogenase. So it takes NADH, converts to NAD and hydrogen ion. Converts to hydrogen ion and NAD. So let's just say there you go. That's what we did. Now, if you keep pushing the hydrogens here, that means you will have overall hydrogen will accumulate here, right? A lot. That's all you're trying to do. So you grab this hydrogen from where? From the glucose molecule. Okay. The second one, number 2, is known as Succinate Dehydrogenase. It takes FADH, FADH2, which is produced from fumarate reaction. Okay, from fumarate becoming malate. Okay, from the fumarate reaction, which should be here somewhere. I don't have enough. So 2-FADH. So we'll take these 2-FADH and takes the hydrogen and pump it here, and then it will convert to FAD. Okay, so produce in the process FAD. FAD. I have no room, so I have to make these weird lines, but hopefully you can think. At the end of the day, what did we just do? We created a proton gradient in the intermitochondrial membrane space. The whole purpose of that was to get the hydrogens from the glucose molecule and accumulate them where? In the intermitochondrial membrane space, the space between the two membranes, so it can drive the pump. Okay? So the hydrogen we took, but you know that energy cannot be created nor destroyed. It can be only converted to one form to another form, from one to another form. So the hydrogen we did, and the hydrogen will come back here. And what are we going to do with this hydrogen? 6 carbon, 12 hydrogen, and 6 oxygen. The carbon and oxygen are used as carbon dioxide, and they're wasted. Just the mechanism works that way. There's no perfect system in the body. The hydrogen we took what? We took all these hydrogens to drive the energy pump. But when this hydrogen comes back to mitochondria, they're toxic. See, hydrogen protons are toxic because they will increase acidity. And that's when we need what? Molecular oxygen from breathing. And then you bring this oxygen from breathing. This oxygen is then combined with this hydrogen to make what? Water. So if you don't have oxygen, eventually what will happen to the proton in this area? They build up. And let me tell you, if you have 1,000 protons in the intermembrane space and they all come in here, you will have 1,000 hydrogen on both sides. That means there's no transport anymore. Because it's simple diffusion, it wasn't pumping, right? Proton are simple diffusion, so if they're equal on both sides, this pump will no longer work. You will not be able to make ATP. This is why we need oxygen to remove the hydrogen. So we create a gradient. So oxygen continuously removes hydrogen, continuously. So it's always low on the inside of the membrane and high on the outside. Always. So this is why we need oxygen to breathe and to make ATP. This is how you make ATP. Of course, if you don't have lack of glucose, lack of ATP. If you use cyanide poisoning, for example, it will block cytochrome P4 cytochrome oxidase, which is the number 4 one right here. It blocks it, and then again you can no longer make ATP. You can breathe all you want, but electron is not transported from here all the way to give it to the proton. If you cannot do that, again it will cause problems.
So 1 and 2 pump hydrogen out. What do 3 and 4 do?
3 and 4 transport electron that you grab from those protons. See, the hydrogen that you get from NADH, this is hydrogen ion. Yeah, so you have to remove the electron from them and later on donate it so you can make a molecular oxygen converted to what? Ionic.
So
Yeah.
does oxygen also power the electron the electron transport chain?
Well, without it, it will stop. See, the transport of electron depends on the oxygen grabbing it from them. Yeah. So without oxygen, the whole thing will stop. Yeah. Okay. Yeah.
So, if the electron transport chain, the job is to pump the hydrogen, we call them electron.
Because it grabs those electrons from hydrogens and other molecules and transport them across all the way to give it back to the oxygen and hydrogen to make water. So it holds the electron tighter and keeps it until the final product comes back. Oxygen and hydrogen come back together. It needs an electron, otherwise it will not make the bond. Okay? Yeah. So it's called electron transport chain because it grabs the electron from what? From hydrogen ion. See, hydrogen is, so this thing called hydrogen has one proton, one electron. We're going into chemistry now. It's just for your understanding. Nobody cares for it in medicine. One proton, so all molecules, all elements should have a proton and neutron in the center, and an electron orbiting outside in an orbit. Proton is positively charged. Electron is negatively charged. Naturally. There's no reason for it. We don't know why. Neutron is neutral. So when you have a hydrogen molecule or a hydrogen like this, it has one proton, one electron, so it matches. It's neutral. But when you move the electron from it, what happens to it? It only becomes positive. So you can call this actually proton because it has no electron. It has only one proton. So to make it back and react with water, you have to give it the electron. So the electron is carried in this fashion. It's used just to create that proton gradient. Okay. But anyways, what is the importance of this? It's not so much the organic chemistry. It's not so much the chemistry of it, inorganic chemistry. What is important is what happens to glucose. That's all. Glucose becomes carbon dioxide and water and ATP. Glucose becomes carbon dioxide and water at the end of the day, nothing more. So if I were to do this, we take glucose, which is C6H12O6. It's used by human cells, which is known as aerobic respiration. Aerobic respiration. So this process moving at this side is known as aerobic respiration. It's known as aerobic. Why? Because it requires oxygen. It's respiration. Why? Because you respirate or inspirate oxygen. And the product will be carbon dioxide plus water. You can balance it. So there should be a total of 6 carbon. Let's say 6 carbon dioxide are produced. and then that's 12 oxygen here. And then how many hydrogen should be here? 6. Yeah. So 6 oxygen, that's 18. So you can say 9 or 6 there, sorry. So that's 6 plus 6. There you go. You can balance it. It doesn't matter. That's not an important part. But in this process, you made ATP. You simply took carbon dioxide, simply took a glucose molecule and used it to make ATP. But in order to make ATP, you have to break it. Okay, and you consumed it and converted to what? Carbon dioxide and water. Water is good for us, no big deal. Carbon dioxide is toxic, we have to breathe it out. Now this is known as aerobic respiration. Who does that? Animal cells, human beings. So without plants, we cannot live. Without us, plants cannot live. So we're dependent on each other. And then vegetarian says, oh, what, we eating eggs and animals. This is more cruelty to me because it will cause human distinction or extinction. But anyhow, at the end of the day, when we talk about ATP synthesis, please memorize few enzymes. Okay, they react in their products. Glycolysis. So there's some enzyme known as glycolysis. So we want to memorize some here. And we need to know enzymes of the Kreb cycle. And we need to know enzymes of electron transport chain. That's all. And by the end of it, we should be getting ATP. Okay.
Now the enzyme between synthase and isocitrate dehydrogenase.
Mm-hmm. Mm-hmm.
That doesn't include with the doing the ATP.
Yeah, Sam.
Remember in GI you want to know how many ATPs produced in the cycle do you want to know now?
I'll go over it. Good, you reminded me. Number one, glycolysis only occurs in the cytoplasm. This is important to know. Glycolysis or anaerobic glycolysis specifically takes place within the cytoplasm. Kreb cycle, mitochondria. But it's within the matrix of mitochondria. Electron transport chain, mitochondria. But where in the mitochondria? Inner membrane. Very good. The purpose of glycolysis is to provide you pyruvate. It gives you pyruvate, 2 pyruvates, and 2 and ADH. Very good. Okay, that's what glycolysis will do. If you have oxygen, you can use this pyruvate to make ATP. If you have no oxygen, it will give you what? Lactic acid, 2 of them. Okay, and 2 NAD. 2 NAD. Rise it now. 1 NADH gives you about 3 ATPs. 1 NADH will give you 3 ATP. 1 FADH will give you 2 ATP. Okay, it's rounded number. You know, 1 NADH technically gives you 2.5, and 1 FADH 1.5, but it's rounded numbers. Okay, how many NADH did we use? And we produce 2 from glycolysis, 2 from pyruvate dehydrogenase, that's 4, 6 from Kreb cycle, that's 10. So 10, how many ATPs are you going to get from NADH? 30 ATPs. Excellent. 30 ATPs. So most of the ATPs coming from where? Oxygen. Because pyruvate dehydrogenase in Kreb cycle depends on what? Oxygen. Whereas glycolysis don't depend on oxygen. So you're only getting 2 ATPs from there. And the 2 NADH are wasted. Okay. FADH also depends on oxygen. So if you have total 2 produced, how many ATP would you get? 4. Okay. So far 4 from 30 from NADH, 4 from FADH, 2 from glycolysis, right? 2 ATPs, remember that? The ATP that's produced by, what were the enzymes in glycolysis that produce ATP directly? Do you remember the enzymes? Pyruvate kinase, that's 1. It produced 2 ATPs. What was that one before that? Phosphoglycerol kinase. And it produced 2 as well. Right? Very good. So let's measure ATPs now. You know what? Let me just erase this. Let's measure ATPs. I just don't want to focus on things that are irrelevant, honestly. I don't care for how many ATPs. And my eraser is agreeing with me it's not working. Okay. Thank you for being nice to me. We want to see how many ATPs can be produced total. Glycolysis, hexokinase used, I'll put negative ATPs. How many of them? 2. Okay. 1, you're right, sorry. 1. Used 1 ATP. And PFK, 1 used 1 ATP. So overall we used 2 ATPs. We lost 2 ATPs from a single molecule of glucose. And then we had phos have 36 ATPs, right? 2 from glycolysis, we said.
And 2 from pyruvate to acetyl. And 2 from pyruvate to acetyl.
Yeah. Now, there's the thing. No, that doesn't produce ATP. I'll tell you which one, yeah. So the energy that's produced directly by enzyme is known as substrate level oxidation or phosphorylation. Substrate level phosphorylation. Please remember that. And there's 2 ATPs produced. Actually, it's not ATP, it's GTP, but it's the same thing. From succinyl-CoA to succinate. No, succinyl-CoA, synthase, gives you 2 GTP. Okay? Succinate, yeah. Now, how many ATPs do we get? 38. From a single molecule of glucose. Okay. We are able to produce ATPs now by two processes. One is with the help of oxygen, and that's known as oxidative phosphorylation. Oxidative phosphorylation. Oxidative phosphorylation produces 34 ATPs. This is oxidative phosphorylation, produce 34 ATPs. FADH and NADH, oxidative phosphorylation. Substrate level phosphorylation. Substrate level phosphorylation produce only 4 ATPs, and 2 of them were glycolysis.
Substrate?
Substrate level, yeah. Substrate. Substrate. Substrate level phosphorylation. And we have oxidative phosphorylation. So by far the most important one is what? Oxidative phosphorylation. Oxidative phosphorylation. Okay, oxidative phosphorylation. Do you remember when we talked about hypoxia and we said it's oxidative phosphorylation that's effective? Okay, the enzyme that you need to know. Enzyme of glycolysis, there's 3. You must memorize them. The irreversible step. Hexokinase 1. You have to memorize that. Phosphofructokinase 1. You have to know that. Pyruvate kinase. You have to know that. That's enzyme of glycolysis. And then in the Kreb cycle, and then we have Isocitrate Dehydrogenase. Then we have to know something alpha ketoglutarate. Usually it's abbreviated alpha KG, which stands for alpha ketoglutarate. Okay, so alpha ketoglutarate dehydrogenase. And you should know malate dehydrogenase. I think that's enough. If you know anything between them, that's no big deal, and it would be a good idea. Now, by far, the most important one out of all of them is isocitrate dehydrogenase. Isocitrate dehydrogenase. Good question. I don't want to tell you. I'll tell you. Because it involves lipid synthesis as well. So I'll tell you. Electron transport chain, ATP synthase, and NADH dehydrogenase in cytochrome oxidase. Okay. Nobody cares for those, whether you know them or not. But this is now. There's one important enzyme that connects glycolysis to Kreb cycle. Pyruvate dehydrogenase. Oh my God. You have to memorize it. You should know these enzymes so good. You should know exactly what is being used and what is being produced. So what do we need to know about this enzyme? Know the reactant. What is a reactant? Substrate, right? So you should know the substrate, which is whatever is being used, called the reactant. Reactant. You should know the product of each enzyme. And you should know the regulators. Regulator of each enzyme, and I'll tell you those. Okay, the regulator. The regulator could be hormones, could be vitamins. Okay, so let's say I'll just include in here vitamins too. So what are the vitamins that we need to know? Okay, for hexokinase, again, all of this is in your book, so it's no point of me writing it. It takes too much time for no reason. For hexokinase reaction, the reactant is glucose and ATP. Glucose and ATP. So let me give you one. Now, glucose is converted to what? Glucose 6-phosphate with the help of hexokinase. And it requires ATP. So the reactant is ATP. And the product will be ADP. plus glucose 6-phosphate. So whatever is you know what? I understand. If you take 6 carbon sugar, known as glucose, and you add a phosphate to it, you'll get the glucose 6-phosphate. So and now I know the reactant in the product. In the enzyme, I can understand too, it's hexokinase. You're like, but where do ATP? See, the source of phosphate is ATP. The source of phosphate is ATP. So you should automatically be forced to think that this reaction should require ATP and should produce ADP. ATP, let's say, it has 3 phosphate. That's ATP. 1, 2, 3. And ADP stands for diphosphate, 2 phosphate. So 1 phosphate was donated. To who? To this glucose molecule. Okay, so triphosphate, diphosphate. If it's AMP, monophosphate. Okay. Now, for this reaction, there's not much cofactors involved, so let's ignore in that sense. Phosphofructokinase. Now, I'll tell you some stuff, and then please remind me this, because what I want to talk about is these things. What is the role of the following when it comes to these reactions? In terms of hormone, we want to know what is the role of insulin, what is the role of cortisol, growth hormone, glucagon, and let's say thyroid hormone, in this case T3. So that's in terms of hormone. We also want to know what is the role of vitamin B1 when it comes to this. Okay. The role of vitamin B1. What is the role of oxygen? Okay, and what is the role of NAD plus or NADH? And let's just say NADH as well. NADH. Okay, and what is the role of enzyme known as coenzyme A? Okay, so coenzyme A. Anything else? I want to make your life miserable. Tell me some more. Beautiful. Biotin will keep. I'll say it right now, but it's not right now. It's actually making glucose more. The opposite. Is biotin known as a vitamin?
Yeah.
Which one?
B6.
Not 6.
B7.
Yeah. 7. What's folate? 9. Okay. Yeah, so I think sometimes students confuse these two, biotin and how many planets are there? Write this down? Okay, if you wrote it down, that's good. I'll take a quick break and I'll come back. One by one, you have to tell me what I wrote because I'll forget.